Hey everyone, welcome to Metafluidics. It's really excited to see you all here. My name is Susan. I'm an undergrad at at MIT. I work at the Media Lab along with most of the other presenters here. And today we're going to talk to you about Metafluidics, which is an open repository for microfluidic systems. So we're super excited to get started, and yeah, let's begin. So I will start off by just briefly discussing our agenda for today. So we're going to start with a quick introduction section. I think there aren't too many people in this call, so we will probably have time to go through everyone, just a quick self-intro, and then we'll go into a discussion on what is microfluidics for those of you who may be unfamiliar with this concept. And then we'll talk about what is metafluidics, which is the repository that we're introducing. And then we're going to have a brief walkthrough of the Metafluidics website, show you how to navigate the site, how to add a part, and have an initial Q&A session in case anyone has any questions on that front. And then our guest speaker, Krishna, is going to introduce the 3DEV Metafluidics design interface. And then we'll just wrap it up with a final Q&A session, and that'll be it for today. So any questions so far on the agenda? Okay, nope, and feel free to ask any questions through the chat as well. We'll have people monitoring the chat and stuff in case I don't see you raising your hand. All right, so let's just get started with an introduction then. So first of all, here is our team. So yeah, let's just have everyone on our team introduce themselves. So Joseph, do you want to begin?
Yeah, sure. Hi everybody, how's it going? My name is Joseph and I'm from New York. I am also one of the undergrads working in the Community Biotech Initiative. And yeah, I'm super excited to be here. Janice?
Hi, also an undergrad here. My name is Janice. I'm also calling in from New York City, but am working with CBI this fall. Yeah, I'll pass it over to Aneesh.
Hi everyone, I'm Aneesh. I'm a high school student based out of Massachusetts. Yeah, that's where I'm calling in from. Lizi, go ahead.
Hi, I'm an undergraduate at MIT. I'm also calling from New York City, but I'm originally from Georgia, the country, but I also worked with Community Biotech Initiative this summer. Susan?
All right, like I said before, my name is Susan. I'm also an undergrad at MIT, affiliated with the Community Biotech Initiative at the Media Lab. And I'm actually calling in from Shenzhen, China, where I'm taking a gap year to do an internship. So it's morning here for me, and yeah, really great way to start off my day. All right, thank you everyone on the team for introducing yourselves. So now let's move on to just a brief self-intro from people in this call. So maybe just say your name, your where you're calling in from, affiliation, and then maybe one thing you're hoping to learn or a device that you're excited about. So let's just make this pretty quick. And while everyone's introducing themselves, if you all can please fill out this Google form to check in. If y'all can please fill out this Google form just so we can, wait, can you guys hear me? It says I'm muted right now.
We can hear you.
I can hear
Okay.
you.
Okay, cool. Yeah, but yeah, just please fill in this Google form to check in, and yeah, someone will send this link in the chat as well. And awesome. So let me just go ahead and start calling on people. I'm sorry if I'm putting you on the spot, but yeah, on my list, let's go with Cecilia. Would you like to go first?
Hello, I am Cecilia Vilca. I am from Peru, and I am an artist working in the borders of art and science. And well, I want to learn only this topic.
Great, thank you, Cecilia. Next, how about David? Hi, David. Can you hear me? Okay. If it's not like, if it's not possible to speak, like feel free to type in the chat. Oh, I can't hear you, David, actually. So why don't you just introduce yourself in the chat, actually? And actually, all right, how about Chris? Chris, do you want to go next?
Oh,
Yeah, thank
sure. Can you hear me?
Yeah.
Okay. So, hi, I'm Chris. I'm currently in Orlando, where I have been leading the closet in a makerspace. But I'm moving to Indiana in a week, where I'm really hoping to found a new lab and hopefully hook up with some of the Chicago spaces for some collaborative work.
Awesome. Thank you, Chris. Next on my list, we have Anders.
Hey there, my name is Anders. I use they, them pronouns. I'm coming from Seattle, Washington on Duwamish land. I work in a biomedical research lab called RareSite and then also volunteer with King County Equity now in the Duwamish Solidarity Group. And I've never really been exposed to microfluidics outside of some like just personal research. So I'm really excited just to jump into this world and see what it has to offer.
Great, thank you, Anders. Next we have Derek.
Hi, my name is Derek Jacoby from Victoria, BC, Canada at the Victoria Makerspace. And I've looked at the Metafluidics project, but not for a couple of years. And so I'm really just wanting to check in and see where the project has gone.
Great, thank you. Okay, next we have George.
Yeah, hello, my name is George, or Georg Dremel. I'm originally from Austria, but I'm based in Japan, in Tokyo, so good morning. So I'm running the BioClub Tokyo, and I'm curious, how can we bring microfluidics to a biohacker space?
Thank you, that's very cool. Next we have Eugene.
Hi, I'm Eugene. I'm over at GenSpace in New York City. I've worked in a couple different labs now, and one of my labs was using LCMS. And back then was when Theranos came out, and everyone got really excited about microfluidics. So I saw your name on the last thing for tonight's talk, so I want to kind of see what this project is about. I love open source hardware and software and science, so I think this would be really exciting.
Thank you. Okay, next on my screen I have, your screen name says OSN_Scott.
Yes, hi there. I'm Scott Pownall from Vancouver, Canada, Open Science Network. That's the OSN part. And yeah, I actually just got an email from somebody in my city that wanted to donate equipment. And I asked what it was, and she said, no Patrons too. Woohoo! So sorry, I'm distracted. Yes, I actually, way back in 2016, I did the How to Grow Almost Anything course, synthetic biology course with David was actually orchestrating that. And one of the sections was on microfluidics, and he introduced the Metafluidics website. So I've always been interested in it. In that homework, I had to get some PDMS to make some. So I was making what I called microfluidics, and I did things like the cell counters for counting cells, where it has the etches on it. So I put some liquefied silk onto it to be able to get the imprint, and then was trying to use that to create microfluidic channel, or just get the imprint with PDMS of microfluidic channels. So I've always been excited about microfluidics, and yeah. So
Great, great. Thank you, Scott. All right, next we have Eldie.
Hi, Hi, my name is Ellie. I'm Peruvian, but now I'm based in California. I'm from UC Davis. I just graduated from my master's and I work in interactive organisms lab at the Department of Design at UC Davis. And I've been working with biosensors. So I'm wondering like how to use microfluidics like for using biosensors on it. I'm trying to change the substrate. Like usually we use PDMS, but I was thinking a bio-based material that could do the same work.
Cool, thank you. Next on my list is Kalaumari. I'm sorry if I mispronounced your name.
Hello, my name is Kalaumari Mayoral. I'm from Mexico. My affiliation is Tecnológico de Monterrey. Actually, I'm a PhD student. And well, my area of interest is biosensors and also point-of-care devices. So I'm interested in learning about the microfluidics in that sense. Thank you.
Cool, thank you. All right, next on my list is Christine.
Hi, I'm Christine from New York. I'm Austrian native as well, like Georg. And I'm right now Chief Scientist at Biobus, CBI Community Lab, and I'm just here to learn about metafluidics. I have some research background using this in the lab, and I just want to see, like, you know, what you've been working on. So maybe you can bring this back to the laboratory in New York City.
All right, thank you, Christine. Next is Tim.
Hi. Yeah, my name is Tim Dobbs. I am affiliated with Counterculture Labs in Oakland, California. Really, I just, have an interest in microfluidics, and metafluidics sounded interesting, so I came to check it out.
Great, thank you, Tim. And all right, so next, Catherine.
Oh, hey, I'm Catherine from the Lab 3 place in Germany, and I was as a student researcher or a student assistant, I had some projects in microfluidics, and I'm eager to explore it in the open source domain as well.
Great, thank you. Next we have Andy. Andy, can you hear me? Okay, never mind. So actually we're running like a little bit low on time right now. So yeah, I'm really sorry to do this, but can like the rest of y'all like introduce yourselves in the chat instead? I just want to make sure we get through all the content that we have prepared for you guys. And yeah, and we didn't expect, I guess, this many participants. So thank you all so much for coming here. And yeah, the rest of you guys, please introduce yourselves in the chat and feel free to like interact with each other and ask questions in there as well. And for people who just came in, please fill out this Google form just so we can check in. We'll send the link again in the chat. So thank you guys so much. And I'm sorry I have to cut this part short, even though it's really great getting to know what you guys all do. So now I'm going to pass this off to Lizi, who will give an introduction on microfluidics. So yeah, take it away, Lizi.
Yeah, so I'm just going to give a brief overview of what microfluidics is. And just judging from your backgrounds, I feel like a lot of you kind of know about this topic, but I'll just go through it anyway. So yeah, next slide. Yeah, so what is microfluidics? So microfluidics is basically just a technology of fluid manipulation, but the dimensions are in tens of micrometers. So just to kind of give you a sense of how like small that is, like one drop from a pipette would be 1 microliter, one period would be 1 nanometer, and like one strand human hair would be 100 microns. So you can imagine how small the channels in microfluidics are. So yeah, microfluidics has a lot of applications in a lot of diverse fields. So chemistry, biology, medicine, physical sciences, very versatile. Next slide. Yeah, so a lot of microfluidic systems are usually referred to as lab-on-a-chip systems. And they're called that because they're able to integrate entire like biological or chemical laboratories on a small, tiny chip on a very small scale. And they do this by the integration of microfluidic channels and using active passive components like filters, valves, mixers, and many others. And you can see a photo of just like an up-close lab-on-a-chip system. Next slide. Yeah, so why microfluidics? Why would we need this? Why would we use it? With microfluidics, there's a big possibility of performing analysis with very limited amount of samples and reagents. You don't need a lot at all. So this really greatly reduces the amount of reagents you need to use, which is gives a positive impact on the environment. It's cheaper for the lab, and you're able to perform a lot more experiments on with just little material. So it also allows a high level of automation and measurement, so less chance of making small errors, and it's more precise. Next slide. So what are some applications for these tools? There's a lot of applications for these tools, but I just highlighted some in these slides. So one promising one is in cancer therapeutics. So as you can see, this is a paper, but basically we could use microfluidics to identify specific cancer cells, like small microfluidic circuits, and also like destroy these cancer cells. I'm sorry if you could hear my background noise. It's New York on a Friday. Okay, next slide. You could also use this for bioremediation, so proteins, for high throughput protein engineering. This is especially important now when creating like the COVID vaccine. It's really difficult to manufacture a high output of proteins. So this is also a way to combat that through microfluidics. Next slide. And also, I also put engineering the human microbiota cellular communities. So there's a branch of microfluidics called organ on chip. So we could put a bunch of like different organs and mimic the way they, like the environment of our organs in our body on a small microfluidic chip. And one example of this is an artificial gut on a chip to prototype microbial communities. So that's also a very promising field as well. Next slide. Yeah. Now we're gonna talk more about the Metafluidics website and what you could also do to contribute to the Metafluidics website. So I'm gonna turn it over to Aneesh.
Hi, everybody. Before we go into a deep dive of the Metafluidics site itself, I just want to give a quick overview, a surface level overview of what Metafluidics itself is. Next slide. So what is Metafluidics? It's an open source community driven microfluidics compilation where people can upload their own microfluidic parts along with their design files and the hardware that allows users to build each microfluidic device. And on Metafluidics, there's almost every single microfluidic type you can think of, including open thermosilers, liquid handlers, and all of the types that Lizi mentioned as well. It was highlighted in the MIT news block, showing how prominent Metafluidics itself was to the science industry. Next slide. So why is Metafluidics necessary? It's necessary because microfluidics are not utilized as much as they can be due to low quality materials, meaning that before Metafluidics, microfluidic devices were not utilized because they were low quality, low grade, and due to their low quality production, people started to use them less. Because of the low quality devices being more accessible since they were cheaper and easier to manufacture, the high quality devices became inaccessible and people started completely throwing away microfluidics themselves due to only the low quality devices. And since the low quality devices were proving that microfluidics needed to have an upgrade in the availability of the high quality devices, Metafluidics was created. Next slide. So here's a picture of the Metafluidics site itself. It includes, as you can see, all types of metafluidic devices ranging from 32-channel controllers to high throughput yeast microchemostats, which are completely different devices. They have different types of pumps, different types of valves, different types of liquids going through them. Next slide, please. And here's a highlighted Metafluidics post, which includes the design files. You can click on the design files below them. We'll show you in a deeper website dive, but there's a description of the device, pictures of the device, design files, basically anything you need in order to produce the device yourself. Next slide, please. So now we're going to take a quick pause for questions on microfluidics and metafluidics. You can post them in the chat or preferably say them out loud so anybody can answer them.
Yeah, if you have any questions at all, feel free to just unmute yourself and speak up or like send it in the chat. And we're gonna have more like Q&A sessions after this. So it's okay if you don't have any for now. You can save them for later.
So one of the things that I did way back during the How to Grow Almost Anything was, I wouldn't call it microfluidics, I'd call it millifluidics. So it's just experimenting. I'd take a tissue culture plate and I'd use this, this stuff that I got from the local craft store called puffy paint. And I was using a 30 gauge syringe to actually just draw lines on the tissue culture plate. And I filled it up with PDMS. So yeah, I wouldn't quite call it microfluidics, but if anybody here wants to explore, it's a great way. You just need some Silgard, some plastic dishes, and puffy paint. Just wanted to put that out there. It was a lot of fun.
Yeah, that's really cool. Yeah, I think like a lot of the design files in Metafluidics are very much like DIY friendly, and they actually provide some very detailed resources and like bill of materials on like where to find these supplies. And so it's definitely very very friendly towards anyone who wants to try out microfluidics from the community. And Joseph and Janice will go into that in just a bit as well. Cool. Any other like questions, cool comments, things you want to share before we move on?
Yeah, I have a question. It's very cool, first of all. And also that sounds very cool, Scott, also. Yeah, as far as like tooling and hardware required, I saw glass was a material there. I certainly don't have any way to cut or blow glass at my house or in my community lab. But like, so what are the, generally, what are the demands to build one of these things? Do we have to have a full fab shop, or is it like you can 3D print it, or what?
So that's one of the challenges with microfluidics is that sometimes you do like need a full like fab shop, clean rooms, like a lot of really expensive machinery. But there are many devices that have been uploaded to the site that just require like a simple 3D printer or have less like stringent and expensive resource requirements. Each part that's been uploaded has like the information and typically has like tags with what things are necessary. So some like may require a much like higher, much more better access to like the resources involved with like making PDMS chips. But there are many on the site that you can like make just with a simple 3D printer.
Cool.
Yeah, it really varies, but usually if the instructions are like high quality, they'll tell you exactly what you need for them. So there should be like any like surprises if you're like halfway through making and such. All right, thank you. Any other questions? Feel free to send them in the chat as well.
Yeah, I had a quick question actually, just going off of what was just said. So I currently use a nanofab facility to make my devices, but I've been looking into 3D printing, and I'm wondering if anyone here has experience doing that and what kind of lowest resolution for feature size they were able to achieve, because we're seeming, I need to get onto feature sizes that are 20 microns and below, and it doesn't look like that's going to be easy. I'm wondering if anyone's had success doing really fine features with 3D printing. PDMS, but if there's other materials, then that's also of interest.
I think I'd be able to like say, I can't say anything in regards to like the specific scale you can get down to, but out of like all the 3D printers, you should always get a resin printer, like those Formlabs printers, in order to make those the most precise 3D print reliefs that you can. Yeah, like I don't think that if you want like really good experimental results that you shouldn't rely on 3D printing. I think 3D printing is a good way to prototype more than anything. Or if you're using microfluidics as an educational tool, then that's also quite fine. But yeah.
If you don't want to have to shell out just to get like the super like the levels of resolution that you may want, you can also look into like different like subtractive processes, like somebody mentioned laser cutting. There are like commercial services that if you send them design files, they will like laser cut whatever material you order from them. It's obviously more expensive than just buying in bulk and then doing the cutting yourself, but then you don't have to like put down the money to get a laser cutter and like learn how it uses. Learn how to use it.
We also have resolution tests for 3D printers on our website, Metafluidics.
I also tried to use a 3D printer to design some microfluidics, and 50 micrometers, yeah, the resolution is not very well. I think that 100 micrometers should be like a reasonable size. If you go like 50 or 20, yeah, the channels are not very well defined and the figures are not well defined, so the experiments are not performed very well. But if you are over 100 micrometers, I think that it could be an interesting option.
I can say a couple of things about both the glass and the 3D printing stuff. So I'm not an expert in either of these particular methods of fabricating these devices, but with glass, you can basically just do it like a bunch of acids. I can look up what exactly you need for it, but all you need to be able to do is make a stencil of whatever pattern you want, stick it on the glass surface, and then you can essentially etch it with an acid. So that's how they usually do this. The issues with doing that kind of etching is that when you try doing the etching, it's hard to get an even polished surface. So there's like a lot of hassle to that. And that's one of the reasons, like making sure that's all evenly etched is not easy. The second thing is for 3D printing. If you use like the resin 3D printers, they all come with their own resolutions. I believe the Formlabs one says they have about 5, but usually it never turns into something like that. As I mentioned earlier, there are some tests you can do to actually characterize how well your 3D printer is doing, and you need to go from there. Like every 3D printer you have is a unique machine that has its own set of resolutions and own set of things. And this could be purely because you've been using it for like 10 years and the laser is not working correctly or something else is not working correctly. Like there's all kinds of things that can come into play. But you need to characterize your own machine and then figure out what is a minimum resolution you can do with that. If you're trying to do something less than 20 microns, it is usually advisable to just stick to photolithography, because you can't beat photolithography in terms of resolution. There's no other way to do it if you want to go small, that small.
All right, thanks everyone. That was a super interesting discussion. All right, if no one has any, or Does anyone have any more like questions or comments before we move on? If not, and feel free to ask in the chat as well, I will hand this over to Joseph then, who will give a quick demo of the site.
Everyone, so as we've been alluding to, Metafluidics is like metafluidics.org is what this is all about. So here we have our open repository site. All of you guys should go there now so that you can either live follow along or just explore it on your own if you'd like. It's metafluidics.org. Finally, you can find us on Google. The first thing that we're going to ask you guys to do is to make an account. I'm currently signed in, but it is really simple. You click sign up, fill out the very short information, and click register, and you'll get an email allowing you to download parts from the site and interact more with it. You're always able to look at the parts, but you aren't able to get the design files until you register. So I will give everybody a minute or two to do that. It's a little bit shameless, but we want to make sure that if you guys are interested in a part that you see, you can immediately start downloading and messing around with it.
While we're waiting, does anyone want to shout out their favorite part?
Yeah.
Favorite design?
There's this one valve that they call it an LED valve that's an indicator of whether or not fluid goes through. I just think it's a really neat device that's on the site. We posted about it on social media. Yeah, that's my favorite part on the site.
I'm a big fan of serpentine channels personally. I just think they look very pretty, especially if you you view it under the microscope. It's just curves all around. And Janice, I think you mentioned yesterday you liked Christmas tree gradients a lot.
Oh yeah, it's the epitome of mixing channels. You just throw all the serpentine channels together, and especially if you're just playing around with colors, then you have this beautiful gradient of colors coming out of it. So, yeah.
Oh yeah, and to address a question that was brought up in the chat earlier, somebody talked about surface tension. How are we supposed to deal with the fluids? We do want surface tension when we work with microfluidic devices because it allows for really interesting behaviors. For example, if you have blue dye and red dye and you feed them into the same channel, they won't mix until you start making these serpentine channels. So you can get these really cool situations where you have multiple substances next to each other and they aren't mixing, but they're still going through the same channels. So that's just an example of how microfluidics takes advantage of the small scale. So now we're gonna go a little bit through the site. It's basically broken up into the confirmation emails typically take a couple of minutes to send. I can wait if you guys would like to, but I think I'll start just briefly going through different parts of the site so that you can still view them, and then once you are able to sign on, you can start downloading them. But yeah, so on our main page we have our blog, which tells you about some of the cool microfluidic devices, and then in addition to the featured parts, we also have our collection and just the ability to browse parts that have been uploaded recently. One of the cool things about the site is that each of these parts has a tag. So if you are interested in, let's say, synthetic biology, you can you can search the tag, click the tag, and it'll show you recent devices that were uploaded in a related area. As we were just talking about with resolution, here one of the parts on the site is a channel width test. It's not necessarily a part that you would make to use, but it is super useful in terms of finding out the characteristics of your individual 3D printers and devices. So let's say say we were interested in organ on a chip or on a chip type of microfluidics. Here one of the parts on our site that we've highlighted before is organ lung on a chip. Here on each part page there's a description of the part that people upload with it, all the keywords and materials and fabrication technologies required. You can see in order to make this chip, you do need like PDMS and soft lithography fabrication technologies, but some of the other chips may instead require 3D printers or laser cutters or various other things. In addition to the chips themselves, there's also the hardware required to operate them. You need like the pneumatic pumps and the microcontrollers in order to run some of them, and like parts will come with them as well. Let me quickly log back in so that you can see the area to download. But yeah, so for example, the 3D printed micromixer, here you can choose between, you can look at the bill of materials, instructions, design file, software, all of those things. In addition to the keywords and also typically publications that go along with it, right? Microfluidics is used in research, and you can upload your papers and like directly look at any paper connected to a part that you're interested in. Another cool feature of the site is that in addition to being able to upload parts, you can also, for people who upload numerous parts, there are collections. So for example, Todd Thorson is somebody that we all very much look up to at the lab, and he has some really cool devices that he's made and uploaded to the site, including the 32 channel controller from earlier, which is like a really complicated microfluidic setup with a bunch of microcontrollers and pneumatic controls. But here he has like the assembly instructions, design files, software, paper to go along with it. And so if you were interested in like making this like really complicated and kind of just a really complicated device, again, one of the issues in microfluidics is that it is very difficult to replicate research done by other labs, because they aren't necessarily going to detail out exactly how everything is set up in their research papers. And so Metafluidics is just a great way for labs to collaborate and upload parts and allow others to do the same research that they're doing. If any of you guys are interested in uploading parts or a collection, we would be like literally so happy. It would be great if you guys are working on microfluidic devices or have things that are related to microfluidics. For example, low-cost syringe pumps. They are great for running, for like powering the pneumatics in a microfluidic device, but they can also be used for like other things, right? You aren't restricted to microfluidics. We had a couple of newly fluidic devices on the site as well. And so anything that you think is related to like metafluidic, like microfluidics, millifluidics, like working on small scales, like feel free to upload them. And if you have enough things to upload, then we will like sponsor you on the site and host a collection for you. Are there any parts that people are interested in searching for or subject areas that people want to look up? All right, if not, I can leave you guys to search on your own, right? No need to do it all together, but again, we just want to stress, if you make an account, then you can download all the things, and it would be great because we can see all of the amazing collaborators and the community that like has been built around Metafluidics. You guys can all become a part of. With that, I'm going to pass it off to Janice so that she can demonstrate how to upload a part to the site.
Great, thanks, Joseph. Yeah, so right now I'm sharing my screen, hopefully. I believe my computer's stalling. Or not. Zoom just sent me a message saying it quit, but oh.
You're still here.
Yeah, you're cutting in and out a little bit though.
Zoom lied, keep going.
Oh, did she disappear? Oh, here,
yeah, I
there
believe
you are. Hey.
Zoom just quit on me, but I'm going to resume screen sharing and hopefully it will not do that again. Awesome. So yes, we are resuming from the Metafluidics site. How you would add a part, you just start by going up to the top bar. There should be a big button that says add a new part. And basically when you reach that button, you'll be forwarded to this page in which you're told how to add a new part, what sorts of information you need to provide. So I've kind of set up this microfluidic chip I've been working on with some friends. it's not complete. I don't have a paper done, so I'm not going to be like talking about it too much in detail. But basically, the most important thing about the name and the description is that it makes your piece searchable. The same goes for these entries below, where you have to insert keywords. So when people search for your device, they're actually able to toggle through these keywords in order to filter through the hundreds of devices that are on the site. So this is a helpful thing to consider while you're going through this, this form. In addition, you can always add keywords that currently don't exist on the site that pertains to your project. Of course, not all of these fields are going to fit to your project. Let's say that you made something that's just for testing or education, then it's not necessarily true that you would have a paper that assists it or that you need any supporting hardware. So you just fill this to the best of your ability, but keeping in mind that you need as detailed as possible, such that if a person picked up all this information, they could recreate your experimental setup or recreate your device in that way. Just a note about what types of files we look for. Often when we do ask for CADs, we like them in common CAD files like DXFs if they're 2D files, or 3D files that are openable in common CAD software like SolidWorks or AutoCAD, things like that. Also, in terms of images, we always like to have a variety of images like a screenshot of your CAD or a sketch of your experimental setup, or an establishing shot of your entire device, such that if you make it your thumbnail, a person gets right away what your device is exactly about. And so you can add those visual files also in this form. Not to mention that once you get to this end part, you'll be asked for a bill of materials. This will have to be in like a Excel sheet file. So we like it when a person lists out what sites they use to get their materials. We like it when we see that a person has to put what costs pertain to each of the materials that they use, things like that. Yes, STL is always really helpful. And so yeah, if you do have a paper published that like connects to this device, then feel free to share that as well. And there are people that have YouTube videos attached to their experimental setup. That's always very helpful. Okay, so I'm gonna forward down to here. So we actually give out licenses to people who upload devices to our website. So these are Creative Commons licenses, and you can learn about like the different types and how they could fit to your device and your intellectual property needs. So yeah, you can always save a part as a draft, just add your part straight up. If you're interested in adding a whole collection, let's say you're in charge of a lab and your lab has made a variety of different devices that you want to make open source, feel free to contact us. Our email is going to be linked at the end of this presentation. We would love to have your collections on our site. But yeah, that's the walkthrough to how to add a device onto our website. I'm going to, are we going to do a Q&A or are we going to forward it to 3Duff just real quick?
Let's do a quick Q&A just in case. So yeah, let me go back to sharing my screen. So before we move on to the 3Duff session, yeah, thanks so much, Janice and Joseph, for doing the walkthrough and the demo. So before we move on to 3Duff, does anyone have any questions really quick on either how to make an account, uploading a part, like adding the design files, anything at all? And we will have another Q&A in the end, so yeah.
I didn't see if there's a patent associated with the part. Do you capture that? Do you ask the uploader to embed that into the database?
You can if you would like to. I think there have been some parts where I found rather than having a paper, they had their like patent attached.
Okay.
Yeah.
So it's not a requirement. So
yeah,
there could be
it's
some
not a requirement,
yeah.
but like it definitely is very helpful for anyone who's trying to recreate your part.
Yeah.
The tags that are that are attached to everything. Is it possible to sort by those categories? Like when you say show more, is that all the tags that exist?
I think those are all of the tags associated with the, when you add a new part, you can like add different tags to it. But as David is pointing out, you can create new tags if you think your part is in a new category of microfluidics that nobody else is up.
I mean more for like searching purposes. Like
for searching purposes, I don't, the tags are specifically for the tags that people put on their parts, so I don't think there are more tags to search by, but you can still search up like words that pertain to any of like the description or the name of a part.
Yeah, like Janice mentioned, the title and the description are all like searchable, so you can definitely search by more keywords other than the tags. Great. Any other questions before we move on to 3DEF? And of course, feel free to send in the chat as well. All right. If not, then I'll give it over to our guest speaker, Krishna, who's going to talk a little bit about the 3DEF design software and its interface with Metafluidics. So yeah, take it away, Krishna.
Hello. Yeah. Can you go to the next slide, please? Okay. Yeah. Hi, everybody. My name is Krishna. For those of you who haven't seen me or heard of me, I'm a PhD student at Boston University, and I moonlight as the executive director for the NONA Research Foundation, which is an open source software foundation for synthetic biology. So as part of my PhD, what I do is I build design tools to automate and simplify the design of microfluidics. And if you can play the video. Yeah. Can you play the video, Susan? Or whoever is managing this.
Sorry, it's telling me my Zoom is crashing.
Ah, okay. Well, it's okay. We don't need the video. I can talk over it. So as part of my research work, I build design tools for, I build open source design tools for engineering microfluidic devices. So what this basically means is I look at how microfluidics engineers actually work on microfluidics, and I kind of like distill what is necessary, like all the science and engineering out of it, and I create automated pipelines. And one of the tools in that, in the vein of those things is 3DEF, which is an interactive design tool, which is basically a hybrid between a normal CAD tool, with which you would draw like the designs of microfluidics, with which, you know, like almost every design you find on Metafluidics will be seen. And the second part of the hybrid is it's from like a PCB designer, where there are predefined components with which you can incrementally build up your entire microfluidic design. So that is what the tool is. And in fact, on Sunday from 4:30 to 5:45, if I'm not mistaken, I'm going to be having a workshop where not only do I show you how to use this design tool for quickly making microfluidic devices, but I will also be going over like the details, like the basics of how to design microfluidics. And then I'll be talking about what the manufacturing processes would be involved with it, and how 3DEF can kind of, you know, like automate some of the manufacturing processes and whatnot. So if you actually come in, there's actually going to be a bunch of different things I'll be talking about in addition to the tutorial, where I will show you how exactly you would build a design from scratch, how to think about designing microfluidics, etc. But also I'll be telling you guys about some of the upcoming community projects, which I'm starting for in the fall, like late this year and in 2021. So if you are either just an enthusiast for microfluidics, or if you're a software engineer who wants to like contribute to some open source code, or if you're a microfluidics expert who wants to add your know-how to like to the 3DEF design tool, that would be something where we'd like you to join in and help us. I can see Kenza has a question for sharing fabrication protocols. I guess I will leave that to the next speaker when they talk about that one. So one of the other things I wanted to bring up during this talk was that in 2021, we are planning to do a lot of integration between 3DUF and Metafluidics. Everything from being able to directly open the designs from Metafluidics onto 3DUF and remixing it, and so that you can save it back in your own new account, to also automatically saving your design from 3DUF onto Metafluidics. So that would be one of the new things that we'd be adding over there. And just to offset what Kenza is saying, because she has a question about the Metafluidics protocols. So one of the things we do with 3DUF is we automate the generation of like the design files, which means that if you need to laser cut your device, it will basically have like a pre-programmed routine about how you need to prepare all the SVGs for your laser cutting, and it'll actually give you the exact design file you need for every single step. I guess we don't add extra instructions along with it, but I guess that should be something doable. And in fact, we also do, right now we support CNC milling and laser cutting. We have like a half-baked version support for 3D printing, but we are actually looking for some folks to help us, like finish up that feature set. So if anybody has questions, I can answer it and answer the, if anybody has questions, I can answer them.
Oh, really quick, Krishna, I managed to reconnect. Do you want me to play the video? I think my Zoom crashed last time when I tried to.
Yeah, sure.
Sorry about that.
Let's see what happens.
All right.
So the video, the GIF you guys saw over there, sorry, the short clip you saw over there is like the design from start to finish of like an entire device, which was published in a paper. I keep calling it the data-dense cell trap. I'm sure it has a more nice, fancy academic name to it. But the thing is, I managed to do that entire design in real time in like 20 minutes. In fact, there's like a 20-minute long tutorial YouTube video where I do this entire design from scratch. And this is just to show about how fast you can be when you're using 3DUF to like create the entire design. So one of the bigger aspects is because we use predefined microfluidic designs, it means that you don't necessarily need to know all the fluid dynamics. In some of the questions we've heard were, oh, can this be fabricated by this process? Do I need to care about the surface tension? What are the materials we need to use? So while all of these things are important, if you want to just start designing microfluidic devices, you don't need to know all these things. And 3DUF is a good place for you, for anybody who's new to doing microfluidic design or any other kind of designs to start off with.
Great, thank you so much, Krishna, for introducing 3DUF. All right, now we are going to move on to our next Q&A session. So yeah, if anyone has any questions on 3DUF, on anything Krishna or any one of us can answer, now is the time to ask. And of course, feel free to ask in the chat as well. So this is actually gonna be our final segment. So yeah, we have plenty of time to answer as many questions as we can on metafluidics, microfluidics, 3DUF in general. So yeah, feel free to just unmute yourself and speak up.
Hello. Sorry, I missed the first part of this talk, so maybe it was already addressed. But like, how many users are there now on Metafluidics? I'm curious.
That's actually a good question for Arnav, but I don't know if he has the stats like pulled up because he's in there regularly, in the website regularly for those kinds of numbers. Is Arnav here?
Last time I checked while I was looking through the site, I think we had about 5,000 registered users. So, but those are the people who like signed up. But in terms of like active users, I don't have an exact number for that.
That's awesome. I've been like thinking, I'm sure you all have all been thinking about like ways to incentivize like the use of the platform. And maybe you can think of like there's different stats of like, you know, you can look at citations, or now there's also these new stats that look at how often your work has been mentioned in social media. I'm thinking like maybe there's like ways of linking that to one of those like stats that you can list Metafluidics kind of as a social media website, and then, you know, mentioning your paper on Metafluidics means extra points for that. I'm not sure, but it would be interesting in looking at like ways of incentivizing putting devices on this platform.
I can't believe I forgot to mention that earlier, but actually, so in addition to like uploading papers to Metafluidics, something that we have done in the past and we're just doing all summer is just posting the parts on Metafluidics onto social media. And what we're about to pub it actually after this like final Q&A, but we have a couple of different social media platforms that we've just like used to like routinely like push out like the 3D printed micro mixer on the site or the Todd Thorson collection. All summer, like basically every day we were putting out like publishing or pubbing rather different people's devices. So if that is something that you're interested in, getting your device to a larger audience, that is also definitely something that we can do if you upload your your parts to the site.
Yeah, we've been doing a lot of like social media engagement this past summer. And even if it's a part that's not already on Metafluidics, we've been like just searching through like the newest like lab on a chip published devices, or like other like published papers and stuff, and just like reaching out to those authors and then giving them shout outs, and then basically just like featuring any like cool microfluidic systems that we come across. So I think that's been helping, but like you said, that can be definitely be done at a much larger scale to really integrate this like social aspect of the repository. All right, thank you so much, Kenza. Any other questions or comments, cool devices you want to share? All right.
I have a quick
Joseph.
question.
Oh, sorry.
If somebody else wants to go, they can totally go. But,
oh, no, go ahead.
I was curious to know, for somebody that's like sort of brand new to the fabrication and actual sort of personal development of microfluidics, I was curious to know if somebody could potentially just touch on what the pipeline of sort of approaching microfluidics as sort of a DIY biologist, or somebody that's doing work from their home, and some of like maybe the baby step experiments and stuff they could run on some simpler microfluidic devices.
David, is it all right if we ask you to answer this one?
Yes, I'm so sorry. Can you repeat the last part of your question?
Yeah, it's basically just, I think the last part was just to outline some of the baby steps in approaching sort of some simple experiments and or designs on microfluidics.
Yeah, that's a great question. So, you know, one area that's pretty cool are things like tape-based microfluidics. Those are really low cost, accessible, pretty easy to go with. There's also, people have made some pretty neat, like very low cost kind of modular microfluidics out of Legos. So a lot of these are really good in learning settings. You know, so I think, you know, fluidics, some of these different systems, obviously there's like innovation applications, there are diagnostic applications. Some are very, very sophisticated, things like organ on a chip that, you know, are really for trying to replace animal testing. But, you know, there's, on the simpler side, I think a lot of the kind of diagnostic tests have been pretty cool too. So things that are a lot more similar to like pregnancy tests and things like that, that have similar, that are based on paper. Those are some really common accessible ones. There are also some like spinning disc ones. It's almost like printing into plastic. So injection molding and also plastic based fabrication tends to be a lot more accessible as well. Like you don't need special clean rooms or anything for that, but you're always trading off, as I think that the students have been talking about. If you start moving towards things like that, then you are going to lose out on the resolution that you would get from a clean room, but you can get the benefit of much more rapid prototyping, cheaper access. Yeah, somebody else chime in. Go ahead.
I think another really cool thing is outside of, I'm so sorry, Susan.
Oh no, go ahead,
Okay.
Joseph.
Outside of just the fabrication pipeline, there's also a lot of tools out there that make designing microfluidic parts a lot easier. Typically, people would use things like Adobe Photoshop or like CAD softwares that are designed to like make literally anything, which means they're really versatile, but also like hard to use. There are a bunch of different microfluidic softwares that make making microfluidics easier. For example, if you were interested in making paper microfluidic devices, which are also like much more feasible to make individually, there's a software called AutoPad, which makes it, trying to see if I can find a link to the paper. But it is like a software that allows you to kind of design paper microfluidic devices in like layers, and then if you have like a multi-layer device, it'll also come up with a way to like fold it into each other so that you don't need to worry about alignment issues involved with that.
So I would, shameless plug here, 3DOF would be the right way to do it, but no, so as part of my work, right, so my advisor has a class for computational synthetic biology. So in that, what we do is one of the homeworks is usually a microfluidics homework. So in that one, the way I usually tell, I make that homework, so I can tell you this. I make it and correct it. Yeah. So the best way to start doing any kind of microfluidics is coming up with like a simple idea that you actually want to make and going down that path. This could be a paper you picked up, or just pick one of the metafluidics parts, right? And see if you can recreate it. You can use 3DOF, and in some cases you can't use 3DOF because the parts are not available. So in that case, you can simply use something like AutoCAD or AutoCAD Fusion 360, which is another free CAD tool you can get. So to actually just create your design. And that is how you kind of get started. Fabrication is usually the part which will block you, but I think between all the makerspaces, at least I don't know where you live, but if you live in one of the bigger cities in the West, one of the makerspaces will let you just make whatever you need to do. So that is the case. And in fact, my lab itself is trying to put out a small prototyping service. It's still in like the beta. We're trying to figure, with COVID, all the hiring freezes happened. And so we couldn't add more people to the team. But so we are also planning to start doing something like a small prototyping fabrication service so that you can get cheap thermoplastic devices, which are basically cheap plastic based mill devices. So that was another thing. Nail polish, yeah.
Awesome. Well, thank you all so much.
Kenza, digital microfluidics is not microfluidics. It's its own thing.
Right, very great discussion. Does anyone have any other comments, any cool things you all want to share? Anything at all? I just wanted to like plug modular microfluidics one more time, because those are like my favorite kinds of microfluidics, I'd say. And I think especially like if you look up like plug and play system or like reconfigurable system, they're really cool. Like a lot of sites or a lot of labs actually you can order like the actual parts from them. They could be in the form of like Lego blocks, like injection molded Lego blocks with like the channels already like pre-molded in, and then you can basically just assemble them yourself by yourself and like figure out different like channel configurations. And I worked with a system called Ampli with the MIT Little Devices Lab. And it's basically like, it's a paper fluidic system that's embedded into like 3D printed like Lego like blocks. And you can just kind of like plug them in together and run like actual reactions, whether they're like diagnostics or drug synthesis reactions. And I think for their latest project, they're actually sending these Ampli blocks out to space with the sponsorship of like NASA. And they're trying to do some like pharmaceutical like synthesis out in space. And it's just a really cool concept. I think like if anyone's interested, definitely look that up. And I think they might be like willing to send you some kits as well. Like, so that's like one super cool thing that I've been exposed to. Cool.
If nobody has questions, I'd actually like to ask the people here. I know it probably wouldn't take too long, but maybe it can be a longer conversation. I wanted to ask the audience over here about, like you guys have heard about 3DOF and Metafluidics. You'll probably learn how to make more microfluidics on Sunday because all of you are going to be there, right? So the question would be for me is, what do you guys think of when you think of open source microfluidics? Like, what is your ideal world? And anybody can start. Yeah. If nobody...
Hello?
Yeah, go ahead.
Yes, hi, my name is Ibrahim.
Yeah.
Yeah, my ideal is to connect between in vitro and in vivo. So that gap.
So just tell me, like, what would a day in Ibrahim be with open source microfluidics?
For the say that again, sorry.
Like, what would your day be if there was open source microfluidics everywhere? Like, what would it actually enable you to do? Like, help us understand a little more.
Oh, for example, is for drug delivery, how we will be able to deliver drugs, but not able to deliver cells. If that makes sense.
Yeah, no, that makes sense. Is this related to your research, Ibrahim?
Yes, sir.
Yeah. So for me, just to give you, so it can also be a little more wild. Like for me, I don't do any bio, so I guess I have that bias. For me, like the ideal open source microfluidic world is, I say, Siri, make me a drug delivery chip, or the Ibrahim drug delivery microfluidizer, or something like that, and it just makes it for me. So that's my dream world. So yeah, that would be really cool. Like if something like that was possible. Cell-free manufacturing from Scott. So do you want to talk about it a little more, Scott, about what that world would look like for you?
Yeah, I guess I'm a geneticist by training, so I work with DNA and I work with proteins. And one of the things that we've been interested in is can you produce, at a simple level, proteins independent of the actual cells? So providing all of the ribosomes, so you throw in your DNA. And you have the transcription and translation system in there, so you're producing the protein. So that would be one idea. The other idea, which actually I developed as part of the How to Grow Almost Anything class with David, was basically metabolic pathways where you have small molecules that are being passed from one enzyme to the next, to the next, to the next, until you get the final thing. And so the idea would be that you'd have like a chamber that contains the first enzyme. So you flow the precursor in, and like a Unix program, it makes the modification, and then it gets pumped into the next chamber and then to the next. Some metabolic pathways, if you try to transfer them to yeast, it may be toxic to yeast. So if you do it in some kind of in vitro system, like a cell-free system. There's just a couple ideas that I've had.
Yeah. So if you had like the open source thing, would you like just want to buy it, or would you want to make it yourself? I'm curious to know.
I'm a hardcore DIYer. So
You want to make it yourself, right?
Yeah. Well, no, I think the thing is that you know, when you have off the shelf thing, then your decisions are defined by what's on the shelf. When you know how to make things, so you know, my experience. So you know, I started doing a lot of molecular biology before the modern day kits. So we had to learn all the basics of the techniques. And I think the same thing is true today. You know, people kits, how to expand beyond the kits. So you get constraints. So I really think I'm getting internet connection isn't stable, so I apologize.
No problem.
But yeah, I think there's a lot of opportunity to, you know, if you have a good grasp of how to make the devices or collaborate with people that do, then there's a lot of opportunity there.
Yeah, no, that actually makes a lot of sense. And in fact, so I'm an electrical engineer by training. So anytime somebody says, oh, we can buy one of these things, I'm like, but no, we can make it. I'm like, but no, I can make this. Like, no, we shouldn't do this. And usually that goes into this cycle of where my advisor tells me, no, Krishna, you can't start a new project. So, but no, I completely understand, but by understanding how it is and not have, like, that's actually one of the bigger problems with how microfluidics is done today. You can just buy a thing, and because they only sell a few different kinds of devices, you can't really miniaturize all your lab automation requirements or all your life sciences. So,
you know,
any more people like who want to pipe? Oh, yeah, sorry, go ahead, Scott. Sorry.
Oh, no, I was just going to say here in Vancouver, we have a company, Carl Hansen from the University of British Columbia. He started up a company called Absolera. And what they do is they take blood from people that have been infected by something. This is just one of the things that they do. So say coronavirus, and actually they got a lot of money coming to them for this one. And what they're doing is trying to isolate neutralizing antibodies against the virus using microfluidics. So he's an expert in microfluidic design, and his company is really focused around creating single cell antibodies that target and neutralize different things. You know, they do contract research with others, but they also have in-house projects. So, you know, that's one area where microfluidics seems to be doing really well. So they've got I think they're collaborating with GlaxoSmithKline, and so they have some neutralizing antibodies that are getting worked through clinical trials right now in record time. So I'm excited about the technology. You know, I run a community lab, so I don't have access to the nano imaging, nano fabricating. I actually, through consulting work, I do work at Simon Fraser University here in Vancouver. They have 40labs.ca, which is the nanofabrication and nanoimaging facilities. But I'd have to pay quite a bit of money to actually access their facilities, their clean room and everything.
So just to pipe on top of that, so I know, at least in photonics, which was what I did in my master's, a bunch of like I can't remember which university this is. I think British Columbia. So they had like this one fabrication pipeline where anybody in the community could send in designs that they would make. They'd have like a fixed price and a fixed process they would go through. And so the entire community could like pool in together all their designs, send it to them, and they would just go through that entire process in one go. So they just have the same process for all the designs. So you have to make sure that whatever you use can be fabricated with the same process. So, and that way, as soon as the device is done, they would do some minimal testing to see if it came out correctly or not, and they would send it back out to whoever made the order. So there are ways in which, if we really want to do it, we could do like for the more extreme nano fab requirements.
Yeah, I'm thinking about printed circuit board manufacturing. There's a lot of interest and there's a lot of diversity in what people can do. And so I can see if there are microfluidic fab labs that people connect to, much like how printed circuit boards are. You can do your simple ones, but when you want to get like the multi-layer, blah, 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 with all these little valves and pumps, then, you design it and then you send it out for fabrication. That would be another cool idea.
Yeah. So has anybody had experience using a manufacturer? Also, I have no idea what time it is because Zoom has taken over my entire screen. So if I'm going late, please let me know, Susan.
Oh, no worries. These discussions are really welcome. So I think we can go for a couple more minutes and then we'll wrap things up because we don't want to take up too much of people's time.
Yeah. So has anybody had experience using any microfluidic fab service? Do I hear crickets? Well, in any case, so I'm sure there's more. Oh yeah, so Kansas says chip shop. I guess they have like a set of fixed devices that they will send it out. And Tanu Singh says, no, not yet, hoping very soon. So I guess they're also making devices. I'd love to hear from you guys about what your experiences with microfluidics are. I guess, I don't know if there's a Slack channel for this, but you guys can find me on Slack and message me. I'd love to hear more about what you want. And also because some of my slides are still in flux for like on Sunday. So if there are specific topics you want more information on how to do stuff with microfluidics, let me know so that I can actually include it into the Sunday tutorial.
Yeah, thank you so much, Krishna. Yeah, this is definitely a great discussion. I think that's part of the whole point of Metafluidics is to try to make microfluidics much more accessible in terms of providing everyone with the design files, the bill of materials, and then like a list of exactly what they need so that we can make this process as transparent and open source as possible. So I think this whole discussion is very relevant and it's really interesting to kind of get to know what people's impressions of the ideal open source system would be and how that can be achieved. And we really hope that in the future, Metafluidics can better facilitate this process and just make microfluidics open to all. So yeah, definitely make like a channel for that on Slack. This is definitely a topic that we're all really passionate about. And for the last couple of minutes, let me just share my screen once again so that if y'all can see this, we are going to shamelessly plug our Metafluidics social media sites. So feel free to or please follow us on Twitter, Instagram, if you want to get the latest updates on what's going on with Metafluidics, how we're making microfluidics more accessible to all, and if you want to see any cool devices that are featured. And feel free to reach out to us either on our social media or via Gmail if you have any questions on anything at all, or if you just want us to like feature something like, hey, here's a cool new device that my lab just did or that this other lab just came out with, and we want you to like post about it or something, and we'll definitely like respond and do so, because we're all about trying to just make microfluidics more friendly to people of all backgrounds. So yeah, I'll give you guys a minute to jot down our social media handles. It's just at Metafluidics. And yeah, we really look forward to interacting with you guys further after this workshop. So yeah, this basically wraps up today's session. Just please remember to fill out the check-in form again if you have not done so. So yeah, we'll send the link out in the chat again if yeah, just so we can get like an attendance and see like who is interested in metafluidics. And this is it, I guess. So thank you all so much for attending. We had some really great discussions, really great questions, and it's amazing to see how just how much interest there is in microfluidics and in making this community more accessible. So yeah, please reach out to us if you guys have any questions or anything you want to share. And yeah, we look forward to seeing you all in our upcoming sessions. So yeah, have a good night or good morning. Yeah, bye.
Can I just say in closing, everybody, Susan, can we just give a very, very just like big special shout out to Susan, Lizi, Joseph, Aneesh, Barre, Janice. They've been working so hard to prepare this presentation for you guys for weeks and weeks. And I thought they did such a great job. So everybody show them some love. Susan, amazing job emceeing and holding it down. Aneesh, by the way, is one of our high school students who's been working with us all summer, who did an amazing job as well presenting. So you guys were fabulous. Just great, great work. Thank you all so much. And yeah, just more love for these guys for their great work and more love for microfluidics, everybody. We'll make that channel for you guys so you can continue the conversation there. See you all soon and enjoy 3DUF on Sunday. Take care, everyone.